Okay. All right. I think let's get going. So I want to welcome everyone to this Ocean ICU webinar. And today the theme is ocean acidification and the carbon cycle and feedbacks between the two. I will be moderating the webinar and the questions. My name's Sophie Clayton. I'm a scientist at the National Oceanography Centre in the UK. We've got three great speakers. So Dr. Jan Taucher, who's at Guillemar, Dr. Sarah Cryer from Heriot Watt at Lyell Centre, and Delon Earle from the University of Galway. We'll be touching on several different aspects of ocean acidification, and I'm excited to hear the talks. So welcome. Thank you for joining us. Before we get on with the exciting science, what I'd like to do is go over the agenda and a little bit of housekeeping. So I will talk a little bit about the Ocean ICU project and give you a little bit of background on ocean acidification, but the panelists will tell you a lot more. Then we'll go into our presentations. And we've made sure that we're going to keep time at the end for questions. So please do submit questions or any questions that come up for the panelists to ask. And we'll be using the Zoom Q&A function for that. So you won't be able to turn on your video or turn on your microphone to ask questions, but I will be keeping an eye out on the Q&A panel, and I can ask any questions that come up and have the panelists respond. So one thing to note is this webinar is being recorded. Your cameras and microphones, as I said, have been disabled. And any questions that you post in the Q&A, I will then relay onto the speakers. So your name is not going to be displayed on the video. And again, like I said, for the questions and answers, please use that Q&A function and I will relay the questions. So we're going to keep that Q&A session for the end of all of the talks. So rather than having a question break between each talk, we'll have them all come in right at the end. That tends to be quite an interesting way to have these question and answer sessions because of the interactions that you find coming up between the different talks and between the different science questions that we're going to hear about today. If you have any problems or comments during or after the session, please do contact us. And the email address that you should use to get in touch is hello@ocean-icu.eu. Okay, so now that we've got that out of the way, I'm going to tell you, first of all, a little bit about Ocean ICU. So Ocean ICU is a pan-European project funded by the Horizon Europe programme and UKRI. And it's a big consortium. We have 31 partners across 15 separate nations. The project itself runs over 60 months, so from 2022 to 2027. We're funded through the EU, as I said, and UKRI at 13 million euros. And where's all that money going? Well, we are addressing a particular challenge, and that is the challenge of what happens to carbon that's being pumped into the atmosphere and then into the ocean. So the EU Green Deal requires us to move away from move towards, I should say, operating in a carbon neutral way. But we do have some momentum of carbon dioxide entering the atmosphere. So how does this affect ocean users? We need to compensate for various different effects, direct effects from burning greenhouse gases, indirect effects due to disturbances of the seabed and the carbon that's stored there. We need to understand how large these different effects are and how they interact. How do they operate? And most importantly, what can we do to reduce or minimize any deleterious effects from carbon fluxes? Our ambition as a project is essentially fundamental research-based. And so what we're trying to do is to understand the current state of the carbon cycle to get an idea of what the baseline state is and assess how future changes in climate might impact that carbon cycle. We're aiming to quantify key processes within this system, and they're relevant to those indirect effects that I just talked about, the different carbon storage pools within the system. We then want to incorporate those key processes into models and improve the way that these carbon processes are represented in climate models. Translate those model skills into new tools that then allow stakeholders to better understand how their activities might impact the carbon cycle. And then finally, bring all of this together to improve our estimates of future carbon cycle impacts of different industrial processes, fishing, climate, etc. So you're going to hear today primarily about some fundamental research-based science, but do bear in mind that the goal is to apply that science in the long term. So I'm going to give a very quick background on ocean acidification and some of its potential impacts. So essentially, we humans have been emitting carbon into the atmosphere for some time, and about 26% of those extra emissions are being absorbed into the ocean. So they're going in directly into the ocean. And we want to understand what happens as a consequence of those extra emissions. So when atmospheric carbon dioxide enters the ocean, when it fluxes into the ocean, it goes through an equilibration process. And essentially what happens is the dissolved carbon dioxide reacts with water to create carbonic acid, which then goes through another set of reactions to eventually produce carbonate ions and hydrogen ions. And so the more carbon dioxide that goes into the ocean, the more hydrogen ions we produce, and that then results in ocean acidification. So we are decreasing the pH of seawater. We can see that this is happening. So what I'm showing you here are graphs that show atmospheric PCO2 on the top line and oceanic pH on the bottom. And hopefully what you can see is that as atmospheric PCO2 increases, pH is decreasing over time. And this may seem like a small increase. What we've seen is a decrease, increase in acidification, decrease in pH. We're seeing a decrease from 8.1, which is what you would expect seawater to be, to about 7.9. But that is, in effect, a 60% increase in protons in seawater. So that's quite a big change in pH. This has impacts on marine organisms. So marine organisms have to balance out their internal and external pH. So there's a metabolic cost if organisms need to work harder to do that. And we also know that as pH declines, that has an impact on carbonate concentrations, and many marine organisms are using carbonate to make shells. And so the lower the pH, the fewer carbonate ions in solution, so that leads to a harder time, essentially, for these organisms to create their shells. So we know that there are these biological impacts I've just talked about, and we're now going to move on to our first talk. And so I want to introduce Jan Taucher, who's going to talk about some impacts between the silica cycle and ocean acidification. So I'm going to stop sharing and hand over to you, Jan.
Yeah, thanks for the nice introduction, Sophie. So can you see my screen now?
Yes, it's perfect.
Perfect. Okay, so today I'm going to talk about the impacts of ocean acidification on the silica cycle and diatoms. So actually most of what I'm showing today is from a paper that we published a few years ago. So if you're interested in more details, just feel free also to check out the paper. So as many of you probably know, diatoms are one of the most important phytoplankton groups and primary producers in the ocean, making up roughly 40% of primary production. And they also often form pronounced blooms and play an important role in the biological carbon pump by forming sinking particles. So a big question is, of course, how are diatoms affected by ocean acidification? And actually a lot of research has been done in the past 15 years or so. And these were mostly physiological experiments. So on the right-hand side in the figure, you can see some data from a meta-analysis that synthesized this body of literature. And basically, you can see that the physiological response of diatoms here in the green box is either neutral or even rather positive. So actually, based on this, the consensus was that diatoms are resilient or even benefit from ocean acidification. Now, what has not been considered so far were potential changes in the silica cycle. And yeah, diatoms need silicic acid to build their opal shell. So basically, their fate is linked to the silica cycle and also how it responds to changing ocean conditions. So as you can see in the figure here, that yeah, silica concentrations in the surface ocean are yeah, actually quite low in most parts of the ocean. And this availability of silica in the surface is again linked to the production and sinking of biogenic particles because, yeah, production basically strips out silica and other nutrient from the surface ocean, and these then sink and are transported to deeper water layers where they're yeah, remineralized and converted back to their dissolved nutrient forms, and then redistributed with global overturning circulation. So basically, that determines the large-scale distribution of nutrients and, yeah, also of diatoms and other phytoplankton. So in order to look at ecosystem and biogeochemical responses to ocean acidification, at GEOMA, we carried out several in situ mesocosm experiments in different regions. So basically, these mesocosms are, yeah, large volume enclosures, yeah, that contain natural plankton communities covering several trophic levels. And yeah, because they're closed systems, they allow us to manipulate CO2, for example, and also measure all sorts of things, all kinds of ecological parameters. And they also have the sediment traps attached to the bottom, so we can measure the flux of sinking particles and also their elemental composition. So what we did then, we looked at the silica content of sinking particles by looking at the silica to nitrogen ratio of these sinking particles. And the results here on the left, or the figure on the left shows results from the data synthesis and meta-analysis of several of these experiments. So as you can see, we computed different measures for the response. On the left is basically the effect size, and on the right, the probability density distribution for ambient and ocean acidification conditions. And actually, we found a surprisingly consistent increase in the silica content of sinking particles under ocean acidification conditions. So overall, this amounted to yeah, a 17% increase in the silica content under ocean acidification. Now, how can we explain this? Generally, seawater is corrosive to biogenic silica, so diatom-derived particles dissolve once they are dead and start sinking, and this dissolution rate is mostly controlled by temperature. But when I looked deeper into the topic, I actually found some older chemical studies that show there's also an effect of pH, and these are shown on the figures on the right. So as you can see, at lower pH, the silica dissolution is slower, which is a bit counterintuitive because you would expect maybe more acidic, slower dissolution doesn't fit together. But yeah, in chemistry, maybe many things are not intuitive. Yeah, we found an approximately 60%, or these studies say that there's an approximately 60% decrease in dissolution rate per drop in unit pH. And that number actually matches really well with the observations from our experiment, where we found a 20% decrease for 0.3 units, which corresponds to OA for the end of century. So basically, this tells us that the response we observed to OA is yeah, not because of biological responses so much from the organisms, but it's really driven by a chemical process that then yeah, affects the organic matter produced. So what we did then is we analyzed a compilation of global sediment trap data to see if this effect is also already occurring in the present day ocean. So we did this by looking at the silica enrichment with depth, which you can also see here on the upper right as the silica to nitrogen ratio, which increases with depth. Just because silica dissolves or remineralize more slower than carbon and nitrogen. So normally we see on silica enrichment with depth, and then we checked whether there's a relationship with pH. And actually there was, so as you can see on the lower right, at lower pH, the silica enrichment with depth is stronger because silica dissolves more slowly at lower pH. So yeah, this in fact confirms the findings and the conclusions also from our mesocosm experiments. And yeah, also tells us that the mechanism really plays a role in the silica cycle already on large spatial scales. So that gave us the confidence to actually put this into a global model. So we implemented this pH effect on silica dissolution in an Earth system model to assess the global impacts. And the figure here shows changes in silica. And it's basically a latitudinal section of the global ocean for the Atlantic and Pacific, and the y-axis is depth. So actually what's most important here is that you can see that ocean acidification leads to a strong loss of silica from the surface ocean. And basically this is transferred to the deep ocean where it's kind of trapped and lost from the silica cycle for longer time scales. So there's some variability due to patterns of surface productivity and global ocean circulation. But yeah, generally, you can say that silica as a nutrient for diatoms is decreasing in the surface ocean. And that also makes sense when you think about the mechanism that we discovered that pH or OA leads to slower silica dissolution. So sinking particles just carry silica to greater depths before it's dissolved again to a nutrient form. And this actually has severe impacts on diatoms in the model. So globally, we found a 13 to 26% decline of diatom biomass, just because in many regions, silica is a nutrient that's depleted in the surface ocean. And as you can see on the right, the loss is strongest in the Southern Ocean and in the tropical Pacific. So these are the regions where in the model diatom productivity is highest. And what's also important here is that we look at the net effect of only ocean acidification-driven changes. So this occurs on top of other impacts of climate change, like warming and just generally less nutrient supply. So actually in the model, the absolute decline in diatoms was even almost 50% of their biomass, which is quite huge. And another point I want to emphasize is that our findings completely contradict the conclusions from lab experiments, which I showed in the beginning, that suggest that the diatoms are resilient or even benefit from ocean acidification. So I think one important lesson from the study is also that the expected responses of organisms to OA or any other climate change driver basically can come out completely different on a global scale when you have such unforeseen biogeochemical feedback mechanisms that also interplay with global ocean circulation. And yeah, that's actually all I wanted to say. So if you're interested, there's many more papers that got published on our OA research. So I just put some highlights up here. So these studies look at other processes or different components of the food web. So if you're interested, just check them out or get in touch with me. And that's it from my side. Thanks for your attention.
Thank you, Jan. That was great. And like I said, if you have questions, please feel free to put them in the Q&A and we will ask them right at the end. So we'll move on to our next speaker. Dr. Sarah Cryer is going to tell us something about ocean acidification in the coastal zone.
Thank you, Sophie. So, hi, thank you for having me. I am Sarah. I am a postdoc currently based at Heriot-Watt University in Edinburgh in the UK. But today I'm going to be sharing some results from my PhD, which I did at the University of Southampton and the National Oceanography Centre in partnership with Coastal Zone Marine Authority and Institute in Belize. So generally, when we think about ocean acidification, we're talking about the global ocean. And we know that global ocean, as Sophie has explained, the pH is decreasing. But when we model this into the future, we're only talking about the open ocean and not taking into account what's happening in the coastal ocean. And so my PhD work was focusing in the coastal region of Belize. So the coastal ocean is the interface between land and open ocean, and it's a key component of the global carbon cycle. Yet it's been historically understudied. It supports both biogeochemically and structurally complex ecosystems, which account for 14 to 30% of the ocean's primary productivity, while only covering 7% of the global ocean area and only 0.5% of the ocean volume. Next. And it is where terrestrial-derived carbon and nutrients are supplied to the marine system, primarily through riverine discharge, which in turn influences the inorganic carbon chemistry and partial pressure of CO2, the PCO2 of coastal waters. And it's important to think about how humans have impacted this as they've accelerated the mobilization of carbon between the inland waters, terrestrial ecosystems, and the atmosphere. And this has a potential influence on CO2 fluxes and coastal ocean acidification. Next. And coastal oceans are often dominated by benthic ecosystems, for example, corals, seagrasses, mangroves, or oyster reefs. And these will photosynthesize and respire and therefore produce and consume CO2. So this is the ecosystem metabolism. And so we have all these different sources of CO2 in the coastal ocean, not just atmospheric uptake, but freshwater inputs and ecosystem metabolism. And understanding coastal carbonate dynamics is important for predicting future scenarios and for the ecosystems that inhabit these regions, as well as the millions of people that depend on coastal oceans for both livelihoods and coastal protection. Next. So is this area here that I'm going to focus on, and what is controlling pH, and is it a source or sink of CO2 to the atmosphere? Next. And this work, as I mentioned at the beginning, was undertaken in Belize, and Belize provides kind of this perfect environment to study an example tropical coastal ocean. So Belize is home to the Belize Barrier Reef System, which is the second largest reef complex in the world, consisting of a diverse array of ecosystems within a network of inshore reefs, both sandy and mangrove keys or islands, patch reefs, coastal lagoons, and estuaries, and it covers an area of 22,800 kilometers squared. And it is also a UNESCO World Heritage Site. The Belize Barrier Reef provides a source of both income to a country and is the form of coastal protection to Belize, which is vulnerable to frequent and intense tropical storms, with 60% of Belize's population dependent on its coast and 40% of Belize's GDP tied directly to ecotourism in the coastal zone. So you can see it's a really important area. So my study site is highlighted in this yellow box. So we have Belize City Peninsula, and the Belize River mouth discharges just north of Belize City, and this is the largest river in Belize. It's a very shallow area between the Belize City and the Barrier Reef, and it's mostly covered in seagrass beds and then these mangrove-covered islands, or keys as they're known locally. Next. And so this provides a kind of a schematic cross-section of what we're seeing in Belize, and it's formed of these seagrass meadows and mangrove forests, which are both very highly productive ecosystems, which sequester and store carbon in both their biomass and sediments, but are both globally threatened. They're very much a hot topic at the moment in terms of blue carbon potential and their carbon storage, but I'm interested in what is their role in controlling coastal ocean acidification and CO2 fluxes in Belize. Next. So initially, I want to understand the influence of the Belize River. Rivers are often large sources of CO2 to a coastal environment. They have lower pH than the open ocean. And so we can use salinity as a tracer of freshwater input. So this figure on the left was created from sensor data collected by this autonomous surface vehicle you can see in the video on the right. So it's measuring at about 0.5 meter water depth. And the lower salinity is constrained to just north of Belize City Peninsula, so where we've got the brown and yellowy green, and then further just south of the peninsula as well, where there's smaller river inputs. And by the time we reach the inner islands, the fresh water has dissipated and we're in high salinity waters. Next. But at the same time, we can measure pH, so this is being measured simultaneously with the salinity sensor. And while we have low pH and we can identify the Belize River mouth by the pink, once we have variation in pH across the study area that doesn't correlate with salinity. So this suggests that there's other sources, other controls on pH and CO2 that are not linked to fresh water. Next. And using discrete water samples, I can calculate the flux of CO2 from the coastal ocean to the atmosphere. So in this figure here, everything that is red is a flux from the ocean into the atmosphere, and anything that's blue is a sink from the atmosphere into the ocean. And you can see across the study region, there is a flux of CO2 into the atmosphere. It's highest generally close to land, where there will be a role of fresh water, but it is also happening close to the keys and over these seagrass beds where there's not a freshwater influence. Next. At the same time, I'm looking at the saturation of oxygen. So using apparent oxygen utilization, which is the difference between the oxygen saturation based on the temperature, salinity, and pressure, and how much we're measuring. So again, the scale bar is flipped, but the colors are still red is supersaturated with oxygen, so we're above 100% saturation, and blue is undersaturated. So we can see there's a little bit more variation in oxygen than we were seeing with the CO2 flux. But the key thing is we still have supersaturation of oxygen happening where we have the CO2 flux. So this suggests that there is high rates of photosynthesis over the seagrass bed. There's a production of oxygen happening. Next. So I'm trying to understand where this CO2 is coming from. So here we have a plot of CO2 against salinity. So using salinity as that tracer of freshwater input and the influence of the Belize River, this line, the red line at just above 400 is atmospheric CO2 equilibrium. So anything above that, we're going to be expecting to outgas into the atmosphere. And you can see, as I've pointed out, the Belize River mouth has the highest CO2 as expected. And then this quickly degasses next, where we have lower CO2. And I should just quickly note, so the black is our measured data, and the grey is what we would expect based on physical mixing, so the conservative mixing of our end members, so the river point and the open ocean, or as close to open ocean point as we can get. And you can see that the data lies beneath this, the predicted mixing. So this is suggesting that degassing is happening at a higher rate than would be expected just based on the physical mixing of the fresh water and the high salinity water. So we have this another indicator of degassing, but where is the CO2 coming from? So we know we can see it's being produced, but I'm interested to identify what is it being produced from, what is being respired. Next. So we can use inorganic carbon isotopes to identify processes as well that are happening. So the important thing to take away from here is that inorganic carbon isotopes, or isotopes in general, are on a relative scale. And so for the dissolved inorganic carbon, so our CO2 and DIC, is that as it gets more positive, we have degassing and photosynthesis occurring, and as it gets more negative, we have respiration, ingassing, or weathering occurring. We know we're not having ingassing based on our CO2 measurements. It's not going to be weathering. So if the delta 13C is lower than we would expect from conservative mixing, it's indicative of respiration. Next. And that's what we're able to show here. So again, we're using salinity as this tracer of the Belize River. We have the conservative mixing shown by the grey dots, and our measured data in the black dots. And we're falling below that conservative mixing line, suggesting that there is respiration of organic matter happening. Additionally, data that I've not shown here, we also have production of phosphate, which is suggesting that there is remineralization of organic matter happening and producing the CO2. Next. So I wanted to identify where this organic matter is coming from. So we can use carbon isotopes, this time on organic carbon, so looking at that ratio of 13C to 12C on the organic carbon. On the previous plot, it was the inorganic carbon, and also looking at the carbon to nitrogen ratio. And here I've plotted up the different kind of sources you could get in a tropical environment. So we'll have the terrestrial plants have much higher C to N ratio than macroalgae and phytoplankton, which plot down much lower, and the seagrass versus the mangroves are going to have different delta 13C signatures. Next. And when we plot our data on, we can see that we're not having any terrestrial inputs of organic carbon, but we're having mangrove, seagrass, and a macroalgae phytoplankton signature. So this is suggesting that we've got a marine source of organic matter that is being respired, that is producing this CO2. And this is interesting because when we think about seagrasses and mangroves, we think about them being blue carbon stores, but actually they may also be a source of organic carbon that's being respired and producing this CO2. Next. So to summarize, we know that the Belize River is a source of CO2 to the coastal ocean, which isn't what we would expect, but this is very quickly outgassed and we are not seeing it have an influence across the study area. And this is despite, and so we are seeing it's not having an influence across the study area, but we are still seeing CO2 outgassing, which is this happening on top of photosynthesis occurring, which is likely happening by seagrass. So this is showing that the biology is producing the organic material that's being respired, but is also offsetting some of that CO2 production. So there is a balance happening. And so despite the seagrass role in photosynthesizing and CO2 consumption, we're still seeing a CO2 outgassing to the atmosphere. Thank you.
Wonderful. Thank you, Sarah. That was great. Just a reminder, please do post any questions that you have for the speakers into the Q&A, and we will circle back around to those at the end of Delon's talk, actually. So our next speaker is Delon Earle, who is a PhD student at the University of Galway. So Delon, I will hand it over to you.
Good day. Thank you so much for that introduction. I'm going to talk about how ocean acidification might impact cold water corals and their ability to sequester carbon dioxide. What are cold water corals? Cold water corals are a diverse group of species found in the deep sea from 50 meters down to 3,000 meters. They can be divided into two main types, soft corals and hard corals. In this talk, I'll talk mainly about hard corals, also known as stony corals, which are reef-building species. They are tiny animals related to sea anemones and jellyfish. Unlike tropical corals, they do not contain symbiotic zooxanthellate algae because they live in depths where no sunlight penetrates. Instead, they must feed themselves by suspension feeding on current-transported plankton and other small pieces of organic matter. As their name suggests, they live in cold waters, generally between 4 degrees Celsius and 13 degrees Celsius. Corals can be solitary or live together in large colonies. Reef-forming corals are found in waters where there is plentiful supply of calcium carbonate to make their external skeletons. Reefs are hotspots of biodiversity and are reservoirs of potential novel biocompounds that may benefit the pharmaceutical, medical, and biotech industries. Cold water corals have a global distribution. Zooming into Ireland, we can see that Ireland is an important cold water coral reef location in the North Atlantic. Although cold water corals were first found in Ireland 150 years ago, modern cold water research only began a little over 25 years ago, with the discovery of emblematic underwater hills found in clusters off the west coast by geophysicists during seismic surveys. Further investigation showed that the summits of these hills supported dense covers of dead and living corals. Drilling showed that the hills were composed of carbonate rather than hard rock. The hills, some of which were 350 meters high, then became known as giant carbonate mounds. So the question is, are corals a sink or source of carbon dioxide. Let's examine these processes individually. Corals can be considered to be bioengineers. Individual bush-like colonies coalesce into thickets, cockpits, and banks that are several meters high and can extend to hundreds of meters, over hundreds of meters. Over geological time, banks continue to accumulate material, becoming mounds. The organic matter produced within ecosystems can become buried, effectively sequestering carbon over a long period. Here, you can see bivalve shells attached to buried coral framework in the photo on the right. Carbonate mounds themselves play a role in nutrient cycling, influencing organic material transferred from surface waters. They do this by interacting with currents to strengthen advection from surface to deep, particularly during the winter season. Reef-forming corals use aragonite to construct their skeletons. Aragonite is a mineral form of calcium carbonate, one of the most common minerals found in the marine environment. Aragonite is particularly important for animals that build shells and skeleton. The solubility of aragonite in seawater is influenced by temperature, pressure, and the concentration of dissolved carbon dioxide. As ocean acidification progresses, seawater becomes more acid, which reduces the availability of aragonite. This can make it harder for corals to build and maintain their skeletons. The depth at which aragonite passes from a saturated high concentration to an unsaturated low concentration state is known as the aragonite saturation horizon, or ASH for short. In this map, you can see that the depth of the ASH, aragonite saturation horizon, varies across the world's ocean. This map highlights some regional differences in the ash. In equatorial regions, the ash is generally found around 200 to 500 meters. Temperate regions, the ash is found between 500 to 1500 meters. And in polar regions, the ash can be found at much greater depths, often exceeding 2,000 meters. The implication of shallowing of ash due to ocean acidification will lead to greater risk for impacts on coral reefs. Monitoring these changes is essential to understanding the future for marine habitats in the face of climate change. Velocity of shallowing of the ash will differ from region to region. Fortunately, in Irish waters, corals are safe for the time being, since ash is over 2,000 meters deep. But for how long? Ocean acidification impacts dead corals as well as living corals. The porosity of dead corals increases, leading to crumbling of exposed dead skeletal frameworks, which ultimately results in the coral reef itself turning to rubble. The various components of cold water corals and mounds exhibit different levels of resilience to climate change. We still don't know much about how corals will adapt to life in undersaturated waters, but some experimental studies suggest corals can compensate. However, this comes at an energetic cost and impacts other processes. As already shown, ocean acidification will likely lead to the weakening of dead coral structures, leading to crumbling of coral reefs, reducing available habitats for other marine life. Mounds form over geological time and show cycles of growth when covered with living reef and hiatuses when the reefs die, leading to erosion due to changes in prevailing oceanic conditions. If living corals, reefs cover is removed due to climate change, it is likely that the mounds will become susceptible to erosion from strong ocean currents. So what can mitigate climate change impacts? Can we mitigate climate change impacts? Of course, conservation and restoration efforts and sustainable management practices are crucial to enhancing resilience in cold water coral systems. We should try to promote natural coral resilience by removing pressures caused by human activities such as fishing. The recent Convention of Biological Diversity, COP15, agreed a global biodiversity framework which calls for the protection of 30% of the ocean by 2030. Protecting 30% of the ocean will require the establishment of networks of marine protected areas. But these need to be future-proofed against climate change. We need to identify the areas where corals will persist in changing ocean. Such areas are known as climate refugia. Finally, we need to restore damaged coral habitat. We are involved in an exciting project called Redress, which look to restore vulnerable marine habitat in the deep sea. Our part of the project is to demonstrate the feasibility of restoring coral reef habitats. Damaged reefs take hundreds of years to reestablish naturally. We will install novel 3D printed artificial reefs to help speed up this recovery. I thank you for your attention.
Thank you, Delon. That was great. And actually, I want to say thank you to all of our speakers. I really enjoyed all of your talks. And so now we are opening up the floor to questions, and I'm really glad to see that we already have a few questions submitted to the Q&A. So I'm actually going to kick off with a question that's directed at all three of you. And so we have a question here from Larissa Lorenzi. Sorry if I mispronounce your name. They'd be interested to have the panelists' views regarding marine CDR techniques and impacts, positive and negative, on ocean acidification. So over to you.
Can I say something?
Yes.
It's a good question. So actually at Geomar, we've been running some experiments also for different marine CDR techniques already in the past few years, partly also with these mesocosm facilities that I showed. I mean, I guess it's a difficult question, right? Because in the end, I think most of the optimistic IPCC projections, they actually assume there's a certain amount of CDR. I mean, without it, we're never able to reach 1.5 or even 2 degrees. So it's really important and urgent that we need to do this research also to assess if the different techniques have ecological side effects, for example, or I don't know, unforeseen biogeochemical feedbacks like the ones I showed. So I think it's important to do this research and see which of these are actually an option. And then, of course, there's also a whole engineering part to it, like how to implement these things on global scales, on gigaton scales. And I think in the public discussion, it's often seen problematic because like CDR is seen as an excuse to not reduce emissions. But I think the point needs to be made clear that we need to reduce emissions, but probably we also need CDR to stay within a safe range of climate change.
Thanks, Jan. Delon or Sarah, any more thoughts on that question?
Interesting question. I do think that sometimes marine CDR can push the ideas away from reducing emissions to actually focusing on CDR. And there's a lot of startups happening at the moment where CDR is there kind of where they're going for, where there's money in it, and are they actually going to have any impact on what is the CO2 emissions associated with the production of the CDR, which I think is an important point to consider, whether it's terrestrial based or marine CDR. I just want to tie this into the other question that's been asked for the management of blue carbon habitats, because they could be thought of as a CDR nature-based solution for climate change. And I think while I've shown that CO2 is fluxing out of seagrass meadows, it's important to think if the seagrass meadows aren't there, where's that CO2? There's going to be more CO2, and that importance of having the mangroves, which are likely producing a lot of that organic carbon that's being respired above the seagrass beds. If we get rid of the seagrass beds but still have the mangroves, there's going to be potentially more organic carbon. And so that, while it might sound a little bit negative, what I've said about taking away from the blue carbon aspect of seagrass beds, I think is very important that we protect them. We need to figure out what the accumulation rate of organic carbon is in these different environments, likely in more temperate regions where seagrass beds have seasonality, that there might be an increase in CO2 flux in certain times of year, and taking that into account when we think about climate change mitigation. Just looking at that CDR. Yeah, and I think, there's positive and negative impacts of CDR techniques on ocean acidification. It's just thinking about that whole carbon footprint of the CDR technique in terms of removing carbon.
Thank you both. I think, it's a super complicated question, particularly when you're talking about using alkalinity enhancement as one of the MCDR techniques and then disentangling all of those pieces. I have a question for Delon. So you ended your talk with a really tantalizing look at this project, this redress project that's going to be used to restore these coral reefs. So I guess my question is, how do these artificial reefs speed up the restoration of these coral habitats if you've got this sort of larger scale problem of the dissolution of aragonite?
Thank you. I have my colleague Anthony here. He's going to answer.
Yeah, thanks, Sarah. It's a good question, actually. So part of this project, we mentioned climate refugia, is to make sure that we site these reefs in areas that won't come under pressure too quickly from changes in the aragonite saturation horizon, for example. So we're doing modeling studies, they're called habitat suitability modeling, where we can take into account we can look at environmental scenarios, future environmental scenarios that we get from IPCC, and model those to see where we expect the corals to be in the future. Okay.
Fantastic. Thank you, Anthony, in the background. Okay, I'm going to move on to a question for Sarah here from F. Colhane. So Sarah, do your results have implications for the effectiveness and/or management of blue carbon habitats as nature-based solutions for climate change mitigation? That's a good one.
Yes, so I kind of answered that one partially already in terms of marine CDR, but I think it is important to just understand the full role of blue carbon stores, seagrasses, mangrove, oyster reefs on the biogeochemistry. There's a lot of focus from the organic carbon perspective at the moment and thinking about it storing carbon, but looking at how, yeah, the seasonality, the loss of these habitats is going to change the amount of CO2 being fluxed to the atmosphere.
Great. Ooh, thank you. I'm echoing from someone's screen there. So we've got actually I've got a question for Jan. And so your talk sort of showed an interplay between these local effects of acidification on diatom growth and the larger scale integrated effect on the silica cycle. And I'm curious on what your sort of perspective is on how that then plays into the broader carbon cycle and how how that will be impacted with future climate change.
I mean, so in this study, we didn't specifically look at yeah, like knock-on effects on the carbon cycle, just because, yeah, it's tricky with the model. But I mean, you would expect that if the most important primary producer decreases its biomass by a lot, then it will probably change the food web and biogeochemical cycling somehow. I mean, other phytoplankton groups will jump in and increase their productivity if one group decreases, but yeah, because diatoms are also important, like for trophic transfer up the food web, I think it will affect the whole cycling of organic matter. And also when you think of, I mean, processes in the biological carbon pump, like the sinking speed of particles, it's also controlled by the density of particles. So when you have more silica or it's lost at a slower rate, then this might actually increase the sinking speed. So it might make the carbon pump more effective, or also protect the particles more from degradation. So I think there's different processes that might be looked at or should be looked at. But I think then for these, yeah, to look at the responses at the smaller scales, then again, you need to go back to the lab and run smaller scale experiments. I mean, I didn't mean that these physiological experiments have no use, because that's really important knowledge. We have to know how organisms respond, but it's just like when you have these small incubation bottles, there's just certain processes that you cannot look at, like the ones I showed. You really need like an ecosystem and kind of a vertical, like a water column, in order to look at the sinking particles. So these things you cannot do in small scale experiments, but I think the two approaches should complement each other. So when you see something in these larger scale experiments, then you go back to the lab and like look specifically at this organism or at these processes.
Yeah, absolutely. That scale interplay is so important to think about. We've got time for just, I think, a couple of quick questions. So, maybe three questions. We'll see. So one here for all of the panelists. Other threats for the ocean. What other threats for the ocean does ocean acidification interact with the most? I think this comes under that complicated question again. So maybe just a sort of a couple of quick hitters.
Well, may I tell on here?
Of
So
course.
the threat that it most likely interacts with is temperature, since we find that the release temperature, higher temperatures, there's less carbon dioxide dissolved in the ocean. So that is the interaction that the threat happens with the most.
Jan or Sarah, do you have anything to add?
I mean, I would also say that on the organism level or ecosystem level, warming is really, it's probably an even bigger issue, because like the physiological responses and disruption might be even stronger than for acidification alone. But then, of course, like when these two interact, there might be synergistic effects, so that that the combined effect is even stronger than the individual effects alone. I guess it also depends a bit on the region. So certain regions or ecosystems or like coral reefs might be more sensitive to warming, while other parts of the food web might be more sensitive to acidification. Other way around, actually, but you know what I mean.
It's complicated. Sarah.
I agree with Jan. I was going to say I think warming's probably likely to have a bigger impact initially because, and I guess I'm thinking about warming in the sense that both are happening because of increase in atmospheric CO2, and but it is also going to change vary regionally, organisms, and that's why also we need in more studies where we're looking at multiple threats at the same time. And if we're just thinking kind of chemically, as ocean acidification happens and the change in carbonate and bicarbonate ions, there's also going to be a change in the availability of other ions. For example, like copper is going to increase, the available form of copper is going to increase. So areas where there's potentially more copper, we might have more copper toxicity. So I think it's not an easy question to answer. And there's so many interactions that can happen.
Yeah, absolutely. We do have one more question. It's a bit of a beast, and I think we are running out of time. But I think if possible, we'll type in the answer in the chat on that one. So I think we're at the hour essentially, so I want to finish up. But just a quick plug here, that if you want to interact with OceanICU, if you want to get in touch, particularly if you work in a blue economy industry, if you're a stakeholder, a manager, decision maker, scientist, policymaker, a citizen scientist, please get in touch. Also follow us on social media platforms. We have many updates about webinars like this, and about new results that come out of the project. So we would love for you to keep in touch. So there's a QR code there to sign up, I think, for our newsletter. And with that, I want to thank Jan, Delon, and Sarah again for some great talks. And thank you all for participating and submitting your questions. And I think we will leave it there. Thank you very much.